welcome to another podcast of Your Higher Self. Today I am joined again with Dr Guillon to talk about his research that he's doing on twins currently. So if you haven't listened to our first podcast, we posted it a few weeks ago, and it is on how vision changes how we perceive life. And he went a little bit in about twins. And in this episode, we are going to go really deep into this research. So Jean-Pierre Guillon, say hello and give us a little bit of information of who you are.
Yeah, bonjour, I am Jean-Pierre Guillon. I'm an optometrist, and the last 15 years I've had a great interest in children or individuals with learning difficulties, and it's within that context that I came across quite a few pairs of twins of different type and also triplets. And I starting to have a great interest because it became evident that they had a sort of problems in learning that other group have, and they had a lot of symptoms and signs within the learning difficulty that were present in all the other people. The interest in twins is because they are the ideal type of subjects to study, because there's different type, but really because it's based on their supposedly identical genetics, we can see if they have similarity, differences that may be original through the embryology, or maybe due to their growing up, or maybe to whatever affect them in their life. That's why I had such an interest in looking a bit more specifically on the number of twins that I've examined myself, but also to look in general on what make twins also different from the rest of the population.
Yeah, and so would you like to explain a little bit on how you did your measurements and how you got into it? Because I know that you had you examined photographs of twins from a photographer.
One of the differences that I had seen when I was examining those children with learning difficulties is that it was very difficult for them to have a good and stable binocular vision. The eyes were not working in synchronicity well enough. And one of the things that I had noticed is that many of these youngsters had one eye which was not level with the other. And so we had an angle, and that created a perception problem. They had to turn their head, they had a head tilt, and it was very difficult for them to be able to track, for example, let words across the page. And I had to correct these anomalies, so to allow them to perceive better and read better. And based on that, and again because twins, especially identical twins, were supposed to be the same, I looked at pictures of twins, and using the same technique of measurements to look at whether we had differences, especially in identical twins.
Yeah.
The technique itself was from a photograph. You could put points on the photographs, the center of their pupil, the bottom of their nose, the middle of their lip, and you can have measurement that is first measure the head tilt, which is induced, but also measure the basic difference in height. Because I wanted other people to use other people's photographs, I came across a book by, I think he's Australian, not Australian, from Austria, photographer that become very well known that produced a book of twins with a very, very specific photographic technique where he only photographed the face of people and always in the same way, so they were ideal. And basically, we scanned those images, many of them, of those from Martin Scholler, his name is. Many of them you can find on the internet. And it became obvious, even from the book cover, of twins, that one had level eyes and the other ones didn't. And when we look at all those twins, if I remember, there's about 35 pairs in the book, at least. With the analysis, we could differentiate between one twin and the other solely based on whether their eyes were level or not level.
So you're saying that identical twins are not exactly identical?
It's exactly the main crux of my interest and the research. So I then started looking at in twins and the different type of twins, what are the similarities and what are the differences. And my approach has been that if we find on the face, on the external part of the individual, a variation across the midline, the right side being different from the left side, the idea is that they may have the same sort of differences inside, and by this I mean in their brain structure and in their brain function. And then that will imply is that the laterality of those individuals, external would be being right-footed, right-handed, right-eyed, and internal would be, do we have a difference in the function of the two hemispheres of the brain? And as we'd seen, there was a lack of synchronicity between the eyes. Do we have, in those cases, also a lack of synchronicity in their brain functions?
Okay, so for example, the twin that has the head tilt and their eyes unlevel, what would you notice in maybe their brain or their behavior compared to the other twin?
Good. Now, most of the research in the past, since the 60s, concentrated on handedness, whether they were right-handed or left-handed.
Yeah.
One of the things that my work has shown is that the laterality, I prefer to call, is not regular. For example, we have in the group of learning difficulty individuals a lot of cross-dominance. Cross-dominance, they would separate the head from the torso from the legs.
Okay, yeah.
Because they have three different, they're different functions. So in cross-dominance, we may have a left-footed person being right-handed and left-eyed, and we can have the opposite, right-footed, left-handed, right-eyed. Now that become important because that's what I saw in some twins. We had, like what we call in them mirror twins, where they were the opposite. One was right-eyed and the other one left-eyed, one right-handed, left-handed, and it was a cross. And one of the things that I've been looking for all this time is the presence of this cross-dominance in all the patients that I've seen. And whether that cross-dominance or the difference in their face, which one was the one that matters. I think everything is linked, and that's basically what I got interested into. If I only look at the face, we basically, in fact, could differentiate twins just by the level of their eyes. And it is normal to then suppose that the difference that they have may be also reflected in their brain structure and in their brain function.
Yeah. So would you say that one twin may have some learning difficulties?
Exactly. And everything else associated with it.
Yeah.
If, for example, one twin is fully right-sided and the other twin fully left-sided, we do know that left-sided individual in general have more problem than right-sided individual because that's the world we lived in. Human, we're some of the only primate that should be right-handed, right-eyed, and right-footed because of the differentiation of the control being on the left brain. In cross-dominant people, for example, that are left-eye dominant, it means that the right side of the brain will be more involved much earlier in their development. And it means also that there may be delay in their perception. And so we can look at, first we looked at the mechanical differences in the alignment of the eyes, but also in the neural differences that may occur. The way this is done in an eye examination is that with a system of polarizing test and polarizing glasses, we can in fact separate the input that goes to the brain from the right eye to the left eye. And we have specific markers that should appear black and equal if the input and the neural analysis is exactly level and exactly the same. What we find in the learning difficulty in the cross-dominant people and in some of the twins as well that are cross-dominant, we find there is a lack of synchronicity in the information, which is basically the neural effect between the right eye information and the left eye being transmitted to the left side of the brain and the right side of the brain before they are combined in binocular vision. And that is one of the things that will make their reading difficulty and their tracking difficulties. It may happen only in one of the twins. It may happen in both twins, but in different ways. So that's the level where we are. Now, when we correct them, that's the interesting part, is that when there is a level difference itself, we correct the mechanics with a system of prism, realigning the eyes, and sharpening equally both eyes. These are the mechanics. If after that we still can pick up a difference in intensity of what the patient reports, what we do is then use a system of color filters.
Oh, yeah.
Because the color itself is totally and solely a neural process. And so that's the secondary part of what we do, is using a color to try to regain an equality in the effectivity of the neural pathways between the right and the left. And in some cases it's quite amazing. They immediately can see better, can read better, and that is what we call visual stress. So the interest in twins, and that needs to be still analyzed, is that try to find out, do we have the same sort of mechanism in fraternal twins, which would be like brothers and sisters, in identical twins, which is exactly the same DNA, or also in mirror twins, where one will be right-sided and one will be left-sided. So that's interest that we have at the moment. Still have to do full analysis, both of the full the number of patients that I have, and specifically the twins. The other things is I'm interested in make people more aware and the researchers more aware that we have to move away from just trying to link a feature to the handedness, and we should more link to a feature to the eye dominance, and there's different type, or their contralateral dominance. So it's a much more complex way of looking at it, but only looking at the handedness solely had been really, really detrimental to any advance in research, and I would say the last 40 years.
Oh, really?
Yeah.
Wow. And I know that you've looked also at theories about DNA, and especially I think it's the twins that made you look into DNA. Would you go a bit more into some research that you're doing with that?
Okay. The interest, so we have to look further for a reason for the difficulty that we have observed. So the two parts we have to look at is their embryology, because that's where it starts, and in the embryology, the role of the DNA and the DNA splitting or combining that happen in the normal creation of the human being. Now, that's where it gets very interesting, and it gets also to the realm of gender, as well as in the realm of the twins. It's a combination. Why the gender? Because although the DNA for the gender is only on one chromosome, it's now known that although most people, even if it's 99% of the people on their chromosome may be binary, male or female, there is in fact, and it's been recognized a long, long time by a different population, there's more than one gender associated to this duality only based on external sex appearance. So because we know that there is a variability and a spectrum of genders, and it's based on the sexual DNA part, I believe that there's probably also there is a variability, a whole range of DNAs that may be present in a single individual. Okay, there is many cases that have been known of people that have more than one DNA in their body, and it's kind of what we call more complex a chimera. That means a chimera from the Greek was the one that were made, an animal that were made with from two animal parts being combined. And the same idea may influence a person both in their physical responses to medicine or physical responses just in general, but also on their emotional or psychological responses. The same way as the difference in the gender, some, as far as their brain function, may have some ability or a combination of ability that can only come by a combination of DNA in their body. So if we go back on the way twins are supposed to exist, the basic approach is that we have a clump of cells that split. And if they split early, and it usually is within the first month or so, 4 to 6 weeks, if they split early, we have identical twins.
Okay.
If they split slightly later, an extra 2 weeks, this clump of cell will have recognized right from left, and if it splits then, we'll have a right-handed individual and a left-handed individual. Sometimes things can recombine.
Oh.
That's why we have conjoined twins.
Oh, yeah, okay. Yeah.
And the zone of where they are conjoined will probably depend at what time did they split and the ovum recombined. So it may be through the head, through the hip, which is one of the most common. So when we consider the area where they are combined, they may be two individual, but the area that they are combined will have both DNA. Now, creating a human is a very complicated system, and it's not that efficient compared to animals. It is thought that for every clump of cell that is created, at least half of it do not survive. That's the first thing. And for many reasons, because the combination of the DNAs from the father and the mother do not combine properly. And we see this in Down syndrome. Okay, Down syndrome are the ones that have survived. Most of those that have a difference in their recombination of DNAs, the body rejects them. And usually reject them in the first month, definitely in the first trimester.
Okay.
The second thing is that the survival of a twin fetus is the chances of survival of twins is very low compared to a singleton. And it is estimated that out of 12 twin fetuses in the first months, one will end up being born as twins. 11 will be born as singleton, where the other twin probably would have either died or been reabsorbed by the surviving twins.
Yeah, so that's what they call it, like when they eat the twin. Is that what people mean?
Yeah, so basically it's now known as the vanishing twin syndrome. But we can have that one that totally disappear and is reabsorbed outside, or one which is absorbed by the surviving twins. So that surviving twins will have the two set of DNAs.
Oh, okay.
His and the brother or sister they're supposed to have. It may not be like in the conjoined twins, 100%. In some cases it is. It's split right the middle. And they have features which are recognizable, but most commonly it'll be localized in one area and in one organ of the body, perhaps two. And that organ could be the skin, that organ could be the liver, whatever was developing at the time. Mm-hmm. Now, so that means that we could have, for example, mirror twins, that one would be right-handed and left-handed. If the right-handed one dies, we have a left-handed person.
Okay.
But if we have the left-handed die, we have a right-handed person. Mm-hmm. But it's different from a normal singleton because it may have DNA of the left side. But if we just look at that, because we also seen cross-dominance, all the studies just look at handedness, but because of the cross-dominance, which is very common, we should look at the brain dominance rather than eye dominance.
Yes, yeah.
Otherwise, the data, I would say at least 30% of the data, when we talk about the survival that exists, 30% of the data they may have twin survivors will absolutely affect the final analysis of any study. And I think there has been a weakness in the science, and I would say the last 30 to 40 years. And again, that's why I'm so interested in encouraging scientists to look at that in a much, much deeper fashion. You can do that. Looking at brain function, that becomes more and more common and able now. But now with the advances of DNA that it becomes more common, I think it makes sense to try to find, do we find a different DNA by testing different organs?
Oh, wow. Okay. Yeah.
Now, if we look at it now, just with the DNA technique that exists, the basic one, which has been popularized, we can either have one with scraping. They try to take cells from the epithelium, which are quite complex cells. And there's another technique, they use saliva. Now, the difference being in the saliva, we have a lot more white blood cells. While if you look at the epithelium, we have a lot more of cells that have differentiated and it really is a specialized epithelium. And what I'm trying to look at now is that those two different techniques, are they potentially a very good way to try to find, do we have more than one DNA in one individual that have other signs and other symptoms of duality? I think duality is the right way to look at it, because it should be a duality as far as the gender, but also I would say a duality as far as their mental, emotional, psychological state, and also just in their brain function. Some may be better than others. Now, this is why, again, to look at the brain function is so important, and look at the laterality, because in the past, a lot of the research was negative. They could not demonstrate the role of handedness in many things they looked at. But I do believe it's because they were looking at the wrong thing. You know, you cannot look at the brain function of the ability to think or the way to think somebody by just considering which hand they write with. You have to look at which eye they see with, which hemisphere of the brain they as a dominance. And I think hopefully now the next 30 years or so, perhaps quicker, that will become the norm. And I do believe very strongly that some of the research that was done in the past that showed a tendency of laterality but did not prove it. The research, the approach was right, but the wrong stick was used. They used the handedness and they should have used something else. So I do believe in the future there's a lot of the thing on laterality that was disproved will be proved in the future.
And then with your theories of having multiple DNA, how would you think that somebody with multiple DNA would maybe act, or maybe something that they will show in their brains, or, as you said, also with gender and sexuality?
Yeah. That's the final interesting thing, because if we look at the spectrum that exists, there may be a variability based on time of which part becomes more dominant.
Okay.
By that I mean, if we look at moods, we know we have people that are depressive and hyperactive. We have some people that become ADHD, but not all the time. The same way in these sort of differences or those type of feeling they have, especially about their gender. If that is fluid, there'll be one side taking over for some time and then another side reappearing. And it may be the same with the brain function.
Okay.
If we have those potentially two ability, we may have one hemisphere being more active during depression and one hemisphere being more active during the hyperactivity. Now, what triggered my interest? I had a few friends that were like that, okay? And some committed suicide.
Oh, sorry to hear.
And what is known is the chance of them being suicidal is more when they are in their depressive state, not in the hyperactivity state, not when they are OCD and stuff like that. My original interest was visually by the input, could I force one side of the brain to be used more, the hyperactive one.
Oh.
And the other side being underused, the depressive one. Because if we can do that, we will be able to reduce depression in people that are that way. They go from one to the other. So that was years ago. So, and I believe there's still the potential of affecting the behavior by the visual and auditory input to control a bit better what emotional state they're in. Now, how would that be possible? One of the basic, I was talking about the use of color.
Yeah.
Now, where that is important is that we use a lot in autistic people. And the autism are the one that are hyperactive and their visual input from the periphery affect their central input. And the periphery is all linked to flight, fight, fight. So if we can limit in those people their fear, we limit their anxiety, we limit their depression, because they feel better. And this is one of the things which is reported by the use of color, because basically it dims down the negative effects of the periphery and allow them to concentrate more in their central vision. I do believe very strongly that that will be a very, very good way, because it's quite simple, to treat a day-to-day behavior. We do it already. That my advice is that when we have a meltdown, whether it's autism, ADHD, anxiety, or anything like that, the thing that we do is the individual has to go to their quiet place, and that quiet place should have the right type of auditory information, which is soothing, and the right type of visual information, which is a very specific light, which then they can choose that makes them feel more relaxed, where the anxiety go, where the depression will be limited. And I think that can be extended with the combined use of now the new system, the new technique of brain analysis, because anything which is like this, there'll be differences in the different wave, alpha, beta, or theta wave that the brain itself has and can be measured. Because the quiet ones, if we have the chance to alter the emotional status by auditory or visual input, and I said the idea was to stay away from the depressive states and put them in a more normal functioning situation. And I believe that's basically what hopefully will happen in the future.
Wow. So, wow, this was like a roller coaster of information. You literally covered every single basis. And I know that you wanting to test your own DNA to see if you have those two.
Yes. And in fact, last week I just sent it was last Friday, I think. I just sent the saliva one.
Oh, really?
I already have the one with the scraping.
Oh, yeah.
And I was fortunate to find out that my grandfather wasn't my grandfather. So you got to be very careful.
Oh, really?
Yes.
Oh, did you tell me that?
Did you know?
No.
Yes, my grandfather.
Wow.
Well, apparently I'm English.
Oh, well, okay.
The previous things, the probability of having an English grandfather is absolutely massive. And that was confirmed by the DNA of my sister, and it was confirmed by the DNA of my son and my daughter. And the way I kind of made sure is that he had a sister, and the descendant of his sister do not have any English DNA.
You know what? Maybe one of your DNAs is English. You know what? Maybe your other DNA will come out French.
That's it. The rest is a bit French or Spanish or, Italian, which we knew, but we had.
Well, you did send me a Spanish research.
Yeah, because I keep reading. I read a lot of different language. But that's and now just because it becomes so common, if there is a simple test that can differentiate from cells that we use, the potential ability to pick up, a different in DNA, that would be fantastic.
Yeah, and should we test me too?
Yes.
Let's make me also a science experiment.
Exactly.
And then if we do that, we can also report on a next podcast once we've got research from both yours, my DNA, any DNA that we find along the way.
Absolutely. That would be very interesting because the new, now this technique being available is absolutely fantastic. Because it's available, there's very advanced things which are available, and what is done more and more by those companies is that they try to link the different chromosome to specific disease, which is quite amazing. Okay, so the one thing, if I go back to the combination of what I said before, we do know that facial features, just photograph of the face, allow us already to pick up rare diseases just the way they are.
Really?
Autism is one of them. Fetal alcohol syndrome is another one. But sometimes some very rare disease can be picked up by photograph of the face.
Wow.
But that should be combined by the same way an easy test that may test different secretion, for example, or different organs to confirm this sort of diagnosis. So there'll be objective measurements that may be available in the future. Now, when you know that if you're more likely to get a sort of cancer, like in my family, it's all intestinal, it'll be in the DNA when it runs on that. So that's one of the things that I think will happen quite soon in the future, because it will simplify the care that people can receive.
Yeah. Wow. Well, thank you so much for all of this information, and I think we should really be excited for the results that you're gonna come up with. And like I said, we're gonna test our DNA. We're gonna try some things out. Jean-Pierre is a good friend of mine, so we will definitely be doing some tests on me as well. And then along the way, I will also tell you guys how it's changing me, and how Jean-Pierre may be changing my life or helping me. I'm hoping it's not the opposite.
Yeah, I was going to say that it's part of the miracles I only do on weekends.
Oh, yeah. This is probably going to be a really exciting space for you to keep updated with. And thank you so much, Jean-Pierre, for coming on here again. And I can't wait to invite you here more often.
Thank you very much. It was a pleasure.
Oh, see you guys. And I will see you in the next episode of Your Higher Self.